Hello and welcome to yet another new episode on the Oro-Oto Cerebral Pathway channel. In the embryology series today, we are going to talk about neurulation and the neural crest cells. The neurulation is formation of the neural tube. The notochord induces the overlying ectoderm to thicken and form the neural plate. This neural plate is the neuroectoderm. According to some authors, primitive node is the one that induces the overlying ectoderm to thicken to form the neuroectoderm, that is neural plate formation. This process is neural induction. And this neural induction is possible because of the interplay between two secreted molecules in the region, the bone morphogenic protein BMP and fibroblast growth factor FGF. The ectoderm, under the influence of bone morphogenic protein, its default status would be to differentiate as surface ectoderm. Antagonizing the BMP would actually bring about neuroectodermal differentiation. And there are very many molecules implicated in this antagonization. There is chordin, there is noggin, and then there is this BMP, bone morphogenic protein. By the end of third week, the lateral edges of the neural plate are elevated to form the neural folds. The folds are raised by rotating around a central pivot point overlying the notochord called as median hinge point. So it is because of the median hinge point or at the median hinge point the folds are raised. And as the folds are raised, the midline of the plate gets depressed. This midline depressed region is the neural groove. This process of neurulation will be completed in fourth week, and this neurulation that we are talking about is actually the primary neurulation, because it is not going to give rise to the all parts of central nervous system. It's going to form the brain and upper 3/4 of the spinal cord. The lower 1/4 of the spinal cord will develop from a different process called the secondary neurulation. So after the neural folds are raised, the folds approximate, and the approximation results into closure. So neural plate forms a neural tube. The tube starts closing at the cervical region, and this process of closure proceeds in cranial and caudal directions. Cranial neuropore and caudal neuropore will eventually remain as the cranial and the caudal opening of the neural tube. The cranial neuropore closes approximately on day 25, and the caudal neuropore closes on approximately day 28. So after the 28th day, it can be said that the neural tube is a completely closed tube. The neural tube thus formed gives rise to brain and upper 3/4 of spinal cord. Failure of closure of the neural tube, though, gives rise to neural tube defects, abbreviated as NTDs. While the neural folds are approximating to form the neural tube, cells at the edges of the neural plate do not participate in closure and incorporation in the neural tube. Rather, they undergo epithelio-mesenchymal transformation and move ventrolaterally to form a separate set of cells, the neural crest cells or the NCCs. By this process of primary neurulation, we get the brain and spinal cord up to the spinal segment, sacral second, S2 segment. The rest of the spinal cord comes from a different process called secondary neurulation. Secondary neurulation contributes to formation of caudal part of the neural tube, which eventually becomes the lower spinal cord. And this involves a different process in which first there is a tailbud formation. The tailbud is a structure at the caudal end of the embryo which develops from primitive streak. The mesenchyme and neural crest cells contribute in formation of a solid structure called caudal eminence. And this caudal eminence later cavitates. And this happens through neural tube extension, in which The neural crest cells form a solid cord of cells that extend into the tail bud. And secondly, it happens through neural tube canalization, in which the solid cord of cells cavitate, form an extension of the neural tube, and eventually the neural tube formed as a result of primary neurulation and the newly added part as a result of secondary neurulation eventually communicate, and the spinal cord gains its full length. So the caudal part of neural tube gives rise to the caudal, sacral and coccygeal segments. It begins at around day 28 and probably is over by day 32. Caudal eminence enlarges, cavitates, aligns with the primary neural tube, and eventually the definitive spinal cord length is attained. If the neural tube formation is defective, what would happen? It would lead to failure of closure of the neural tube. It can happen in the region of cranial neuropore, or it can happen elsewhere. The different anomalies, the different spectrum of anomalies possible because of the neural tube failure of closure can range from anencephaly to spina bifida, and to a reasonable degree the neural tube defects are preventable by supplementation of folic acid. So it is important for a pregnant female to have high levels of folic acid, especially during the period of organogenesis of the embryo. Let's just talk something about the neural crests. The neural crest cells are a temporary group of cells unique to vertebrates that arise from the ectodermal cell layer. They migrate extensively and give rise to a diverse cell lineage. Well, this is possible because of their epithelio-mesenchymal transformation. And the various cells they form include melanocytes, the craniofacial cartilages and bones, smooth muscles, peripheral and enteric neurons and ganglia. And because of this wide variety of cell lineages it is able to form, the neural crest cells are also termed as the fourth germ layer. These neural crest cells come with a certain set of characteristics. Like, number one, they have multipotent capability, that is, ability to give rise to several precursor cells. Secondly, they have migratory properties. Neural crest cells break free from the neural folds and migrate to all the areas throughout the body wherever their presence is required. And third one is regulation. It is the ability of the embryo to control the loss of cells by increasing the proliferation of the remaining neural crest cells. So how do we actually keep a track of where all the neural crest cells migrate in the body and what all do they give rise to? This is possible by grouping them into four anatomical regions. Number one, cranial crest cells. They should be present in the region of head neck. Second, trunk crest cells should be present in the region of embryonic trunk. Then we have a vagal and sacral crest cells as number 3, and we have a special group as number 4, the cardiac crest cells. The cranial crest cells, cranial neural crest cells migrate to produce the craniofacial mesenchyme, which differentiates into the cartilage, bone, cranial neurons, glia, and connective tissues of the face. The cells also enter the pharyngeal arches and pouches where they contribute to the formation of thymus, bones of the middle ear, and the jaw. The trunk neural crest cells give rise to two populations of cells. One group of the cells is fated to form melanocytes, migrate dorsolaterally into the ectoderm, and the second group of cells migrate ventrolaterally to form the sympathetic ganglia, adrenal medulla, and nerves surrounding the aorta. Third group, vagal and sacral neural crest cells. This group develop into the ganglia of enteric nervous system and the parasympathetic ganglia associated with the region, and the cardiac crest cells develop into melanocytes, cartilage, connective tissue, and neurons of some pharyngeal arches, and also give rise to regions of heart such as musculoconnective tissues of the large arteries and part of the septum. The neural crest cells are known to migrate to vast regions and give rise to all diverse set of cell population. Neurocrestopathy is a term used to describe any disease related to maldevelopment of neural crest cells, and it ranges from medullary carcinoma of the thyroid gland to neurofibromatosis type 1 to Hirschsprung's disease. Well, this is neurulation and neural crest cells for you. Happy learning.